Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is taken from our spring 2018 issue, and is titled, Bringing Immuno-Oncology Together, How an Immunologist and Oncologist Collaborate. Following on from our last episode around Immuno-Oncology, this episode will be in a slightly different format and will cover a Q&A with two experts, discussing their unique perspectives on the ways in which Immuno-Oncology is reshaping the industry. The experts whose answers and perspectives I'll be going through are firstly Dr. Julia Schüler, Research Director at Charles River, who specialises in oncology, and secondly Dr. Louise Brackenbury, Principal Scientist for Cell Biology at Charles River's newly acquired KWS BioTest, which specialises in immunology. I'll be reading out the questions asked and then their answers in turn. So now on to the main part of the episode, bringing Immuno-Oncology together, how an immunologist and oncologist collaborate. For many years, immunology and oncology operated as separate disciplines within drug discovery. However, recent advances in immuno-oncology, IO, have demonstrated what can be achieved when the two disciplines join forces in developing novel treatments for patients. From checkpoint inhibitors to CAR T cells and model selection designed to reflect more personalised medicine approaches, immuno-oncology is shifting how both immunologists and oncologists approach drug discovery and development. So the first question: what made you realise that immuno-oncology was going to have a major impact in your field? Louise says, "There's been plenty of evidence over the years indicating an important role for the immune system in controlling cancer. However, the difficulty had been finding tractable targets. For example, in a 2006 report, Gallon and colleagues demonstrated that the density and location of tumour-infiltrating CD3+ and CD45RO+ were a better indicator of patient survival than classical methods. What really changed everything was when the first few datasets from early clinical trials started to appear, showing the incredible results with ipilimumab alone and in combination with nivolumab. It immediately became clear that enormous progress had been made, and that our ability to manipulate the immune system has provided us with a powerful tool to fight even late-stage and chemotherapy or radiation-resistant cancers." And secondly, Julia answers, "The complicated crosstalk between cancer cells and the tumour microenvironment, TME, with its different compartments, was a research focus in tumour biology for decades. Nevertheless, identifying druggable targets in the TME remained challenging. Science magazine's Breakthrough of the Year in 2013 summarised the impressive results achieved in clinical trials with checkpoint inhibitors, first in late-stage melanoma. For the first time, specifically targeting a non-tumour cell led to significant tumour cell killing across different indications, independent of staging and treatment history." And the next question: what surprised you when you started to conduct immuno-oncology studies? Firstly, Louise. Coming from an autoimmune inflammatory background, I had to get used to the idea that although we were using the same in vitro assays we had developed to model autoimmune disease, there was now a drive to enhance, as opposed to suppress, inflammation with novel immuno-oncology drugs. In optimizing these assays, we often had to dial back the stimulus to open the therapeutic window. In turn, this meant we had to completely invert the way we were thinking. For instance, where we had been trying to expand regulatory T cells, Treg, we were now testing therapies aimed at inhibiting the same cell type. And secondly, Julia's answer. Coming from an oncology background, mainly developing targeted therapies, I had to get used to the idea that there were no available biomarkers at the time to aid in selecting the model with the highest potential efficacy. Not only did the in vivo model have to be adapted to the new mode of action, it also changed the readout. Besides tumour cell killing, immune cell proliferation, dissemination and differentiation also had to be evaluated in the in vitro and in vivo models. And the next question we asked: what have you learned from the other discipline, either immunology or oncology, that you wish you knew earlier on? Louise answers, "It is fascinating and challenging to understand the breadth of mechanisms by which different tumours evade immune responses. For example, cancer cell immune editing drives the generation of tumour variants resistant to immune cell recognition. The major histocompatibility (MHC) class 1 pathway mutations can allow cancers to become defective in antigen presentation to tumour-specific T cells, and tumour cells can also induce tolerance, apoptosis, or exhaustion in tumour-specific effector cells. Secretion of immunosuppressive factors such as prostaglandins, TGF, and VEGF drive the generation of less effective immune cells, including myeloid-derived suppressor cells, MDSCs, and TREGs. While these effects are hurdles, they also represent opportunities for the identification of novel targets. To be involved in a field with such a wealth of novel targets is unprecedented and is driving the huge interest in reprogramming these cell types, or inhibiting their function, to generate robust anti-tumour immunity." And next, Julia's answer, "It is exciting to learn more about the complex multidimensional interactions different immune cell types can perform. To generate a resourceful immune response while maintaining self-tolerance, the immune system is tightly regulated through a combination of stimulatory and inhibitory signals. How to influence this balance in an immuno-oncological context, and to understand how many different factors play a role in this complex biological system, is an ongoing effort." And the next question. Did you have any misconceptions about the other discipline? What made your thought process change? Firstly, Louise. It took me a while to realise that the revolution in oncology had started, and that there was a key role for conventional chemo or radiotherapeutics, not only in driving tumour cytotoxicity, but also in enhancing the immune recognition of cancers. Although such cases are relatively rare, local radiotherapy applied to a single lesion in combination with ipilimumab has been shown to induce tumour immunity in non-irradiated metastasis, suggesting irradiation of one site can boost systemic immunity. Abscopal effect. Yoshimoto and colleagues tracked tumour antigen-specific CD8+ T-cell responses before, during, and after chemoradiotherapy, and showed that, at least in a proportion of patients, CD8+ T-cell anti-tumour immunity was enhanced. There is now an increasing body of data to suggest that radio and chemotherapy play a dual role in both direct tumour cytotoxicity and enhancing immune function. And next, Julia. I originally underestimated the influence of immunology beyond direct manipulation of the immune system through different modes of action. The induction of immunogenic cell death by well-established compounds, like oxaliplatin, on top of its cytotoxic effect, made me aware of the fact that many, if not all, of the standard of care therapies in oncology have to be re-evaluated in the light of possible immunogenic components of their activity profile. This will help to identify new treatment options and therapeutic areas. And the next question. With the rise of immuno-oncology, how have changes in the clinical oncology landscape affected preclinical model development? Firstly, Louise. Preclinical testing of chemo and radiotherapies was historically carried out using xenograft models, in which the immune system is largely lacking. In immuno-oncology, there is a requirement to have an intact immune system in place, and either syngeneic or humanized patient-derived xenograft (PDX) models have become more commonly used. While syngeneic models are suitable for many small molecule programs, a lack of species cross-reactivity limits their use for the development of many biologics. To overcome this, humanized transgenic mouse models may be generated so that an anti-human antibody may be used in a murine model. When developing such models, it is critical to ensure that the biology of the target is recapitulated, not just confirm expression. An alternative is to perform preclinical testing using PDX in animals with a reconstituted human immune system. This can be a very powerful approach, particularly when screening against a broad range of PDX. A different school of thought revolved around the acceptance that animal models may not be informative, and the rapid growth of complex in vitro 3D culture systems has allowed a major shift towards a lot of preclinical drug discovery in the immuno-oncology space being carried out primarily using in vitro human assay systems. Such complex multicellular systems can model the tumour microenvironment well, and in co-culturing with immune cells in vitro, you can monitor the effects of novel biologics in the tumour microenvironment. And next, Julia. Preclinical cancer research relies strongly on animal models, often based on human tumour cells transplanted into immunocompromised mice. The major readout of PDX models is delay of cancer growth or tumour shrinkage, known as tumour growth inhibition, TGI, which is broadly accepted proof of efficacy for cytotoxic and cytostatic therapies. Immuno-Oncology compounds, however, require additional readouts beyond TGI. Conventional PDX models can only partially reflect the role of the TME, vasculature tumour stroma interaction, tumor immunology, since human tumour cells are interacting with a microenvironment of mouse origin. This gap is likely to contribute to the high failure rate of new cancer drugs in early clinical development, often not meeting their primary endpoints while preclinical data has been encouraging. It is now recognised that the severely impaired rodent host immune system is not always sufficiently reflective of certain aspects of host-tumour immune interaction. Thus, beside new models recapitulating the crosstalk between tumour and immune system, additional readouts are under development that can help to understand preclinical data and have the potential to identify translational biomarkers. And the next question. How has the advent of personalised medicine affected immuno-oncology? Firstly, Louise's answer. Personalised medicine has had an incredible impact, particularly with the use of CAR-T cell therapy in hematological malignancies, and by expanding tumour-infiltrating lymphocytes, TILs, from an individual's tumour in vitro with IL-2, and then reinfusing. Such therapies mean that patients can benefit from highly specific treatments, which provide the potential for far greater success than the current standard of care alternatives. However, due to their cost and complexity, they are only available to a select few. Moving forward, the challenge will be to make these therapies accessible to all. There has been a lot of interest in trying to develop off-the-shelf versions, applicable to multiple recipients at a reduced cost, but this remains elusive to date. The generation of bispecific antibodies, which bring immune effectors and tumour targets in close proximity, also show promise. But each individual's tumour requires costly screening to identify a tumour-specific antigen. This model is also dependent on the recruitment of a functional, non-senescent immune cell, although this may be overcome by co-administration of other therapeutics, such as ipilimumab or nivolumab, or immunostimulatory treatments targeting co-stimulatory molecules, such as OX40. What is most exciting is that personalised approaches are being adopted in cancer treatment. With the discovery of better biomarkers, lower-cost personalised treatments are more likely. And next, Julia. Personalised medicine in the light of immuno-oncology is mostly defined by designing the drug, cells, compound or vaccine, specifically targeted against the individual tumour. As immuno-oncology is based on the idea that the body fights its own cancer, personalised medicine is a fundamental part of this treatment strategy. This requires that the individual cancer is profoundly characterised to identify its unique weaknesses and druggable targets. This elaborate approach implies the possibility of a growing knowledge about tumour biology, which might help us to identify possible treatment strategies tackling common characteristics of specific tumour subtypes in the next 5 to 10 years. In parallel, the development of urgently needed predictive biomarkers will be facilitated. And the next question. What are the biggest opportunities for immuno-oncology to improve patient outcomes? Firstly, Louise. Tumours have been classified as being immunologically hot, e.g. melanoma, or cold, e.g. prostate cancer, based on the degree and type of immune infiltrate. Hot tumours are therefore relatively highly immunogenic, but to counteract this, the tumour has developed a range of mechanisms to subvert this response. Checkpoint inhibitors work relatively well in the context of a hot tumour, as they can reverse at least some of the immunosuppressive mechanisms to unleash the existing T-cell response, but they can be less effective in combating immunologically cold tumours, to which a T-cell response may never have developed. Immunogenic cell death, ICD, is a form of apoptosis caused by chemotherapy agents such as oxaliplatin or radiotherapy. It is characterized by the detection of danger-associated molecular patterns, DAMPs, including extracellular ATP and surface-exposed calreticulin by tumor cells after induction of endoplasmic reticulum stress. Such DAMPs drive dendritic cell activation and promote the development of an anti-tumor CTL response. The careful timing and use of combinations of chemoradiotherapy or oncolytic viruses in combination with checkpoint inhibitors may render cold tumors more immunogenic, and there is a great opportunity for immuno-oncology to improve patient outcomes in both hot and cold settings. And next, Julia's answer. The deeper understanding of how fully approved therapies influence the patient's immune system opens the possibility for an endless number of combination approaches. Well-known treatments can be repurposed in combination, leading to increased patient benefits in a timely and safe manner. The possibility of targeting immune cells or other parts of the TME can prevent the development of acquired resistance against targeted therapies. Beyond that, it becomes possible to treat a tumor already heavily pre-treated, as the mode of action of a checkpoint inhibitor is largely independent of the altered signaling pathways within the tumor cell. The possibility of influencing tumor growth via the microbiome is another opportunity to improve a patient's outcome. Preclinical data is promising, but clearly indicates that, like other strategies in immuno-oncology, the mechanism of action is not fully understood. Finally, the knowledge around the importance of the tumor stroma crosstalk for drug sensitivity, as well as resistance, is becoming more and more obvious. Drugs targeting stromal cells and other compartments of the TME beyond TILs are currently under investigation. And the next question. What are the biggest challenges faced by the field of immuno-oncology right now? Firstly, Louise. In many ways, I think one of the biggest challenges to those in the immuno-oncology field is to determine which combinations of checkpoint inhibitors are best for each cancer indication, and for the individual. The success of ipilimumab, pembrolizumab, nivolumab, and avelumab has meant that an unprecedented amount of money has been put into identifying novel checkpoint pathways and immune stimulatory modulators. There are currently 636 registered clinical trials involving nivolumab alone. Looking forward, therapies targeting a range of other molecules such as TIM3, LAG3, IDO, CD39, CD73, or arginase will come online. As these new therapies reach the market, the potential combinations will increase exponentially, along with the costs, which will eventually be passed on to the patient. We are likely to see increases in the response rates of patients in combination trials, but the key challenge will be in identifying biomarkers that can enable the right combinations to be chosen for each patient's tumor. When you factor in potential combinations with cancer vaccines and different chemotherapeutic agents, this task seems particularly daunting. Further, although efficacious CAR T cells along with other immune modulatory therapies can be associated with significant immune toxicity, managing the balance between efficacy and toxicity remains a challenge, and as patient survival increases following successful treatment, there will also be a need to understand more about how immuno-oncology therapies affect the immune system long term. And next, Julia's answer. Many of the challenges in immuno-oncology go back to the rapid development the field experienced in the last few years. The high number of compounds entering clinical trials, without any reliable biomarker for patient stratification, leads to results that are difficult to interpret. Many of those compounds entered clinical trials lacking complete preclinical data packages. This exaggerates the problem, as many observations were completely unexpected, especially regarding side effects. The delicately tuned immune system in a patient suffering from cancer is difficult to control, and the toxicities are completely different from those of targeted therapies or cytostatics. In contrast to the latter, side effects in immuno-oncology seem only partly dose-dependent. The design of beneficial combination therapies for defined indications or even individual patients is challenging. the lack of predictive biomarkers and the fragmented understanding of the biology behind it lead to disappointing results in clinical trials. And now the final question. How do you anticipate immuno-oncology drug discovery and development changing in the next 5 years? Firstly, Louise. Looking towards the future, cancer therapies will become more specific and targeted to minimize off-target effects. Macrophage-targeted compounds, such as CSF1 modulators, are likely to result in specific depletion of MDSCs and other myeloid populations, while leaving the lymphocytic compartment intact. Whereas Treg-targeted biologics, such as anti-TIGIT or anti-GITR antibodies, may specifically deplete this population through antibody-dependent cellular cytotoxicity. This will ultimately allow a more robust anti-tumor immune response. Our increasing ability to manipulate genetic information and introduce or delete specific genes using CRISPR-Cas9 technology gives us a unique opportunity, perhaps in combination with immunomodulators, to induce neoantigen-specific CTL responses while preventing or reversing exhaustion of the expanded T cells. High-throughput gene sequencing, in combination with proteomic approaches, will allow for more specific analysis of tumor biopsies and determine which combination of immuno-oncology modulators are most appropriate for the patient at that time. As tumor escape mutants evolve and the therapy loses effectiveness, this process could be repeated to identify novel neoantigen targets. Finally, through the use of high-content analysis, the biomarker field will catch up, enabling better stratification and targeting of what will become a suite of immunotherapy combinations to the specific needs of the patient. This is an exciting time to be involved in immuno-oncology drug discovery, and I believe that this field offers real potential to enhance long-term survival rates, particularly in hard-to-treat cancers. And now Julia's answer. Immuno-oncology drug discovery will broaden by manipulating not only T cells in well-described ways, but by targeting myeloid populations to trigger tumor shrinkage. New advents like the microbiome and the combination of treating different parts of the TME are evolving tremendously and are rapidly moving towards innovative therapeutic strategies that could translate into tangible benefit for cancer patients. The emerging field of cell-based therapies will have a remarkable impact on the FDA approval policies in oncology. In the past, approvals were typically restricted to compounds, but the field will be expanded to include medical devices which enable the production of personalised cell-based therapies. This will have an impact on drug development strategies in the preclinical space as well as in clinical trials. The answers in this Q&A article were given by Dr. Julia Schüler and Dr. Louise Brackenbury. Dr. Julia Schüler studied veterinary medicine at the Freie Universität Berlin, Germany, and at the Tierspital Zurich, Switzerland, where she received her DVM. She then joined the lab of Professor Dr. Heiner Feibig, founder of Oncotest GmbH, where she worked on orthotopic implantation of solid cancer PDX in immunocompromised mice. After receiving her PhD from the University of Berlin, Dr. Schüler worked in the field of innate immunology at the Max Planck Institute for Immunobiology, Freiburg. Dr. Louise Brackenbury received her PhD in viral immunology from UCL, prior to 2 postdoctoral fellowships at Imperial College and Bristol University. She joined KWS BioTest in 2012, before it was acquired by Charles River in early 2018. If you've enjoyed this episode, then as always you can subscribe to the journal free of charge by visiting our website ddw-online.com, and you'll need to subscribe soon in order to receive our next issue. If you visit the website, you can also view all of our past articles, including references and images, and download the original PDFs. You can find the links in the show notes. If you're enjoying the podcast, then do leave us a review and subscribe, and you can also follow us on LinkedIn and Twitter. Thanks for listening, and we'll hope to see you in our next episode.